This is aimed at patients and their families, and I'm entirely willing to take questions at any time. We use this set of slides at the Patient Connect meeting in June, and we've updated the slides a bit since then. Okay, great. So, and can I point there? No. Oh, I can.
So it'll be easier if you want.
Easier if I stay there? Okay. So we start with the very basics of molecular biology, which is what are genes, what are what's a chromosome, what's DNA, RNA, and protein. And the reason we start here is because the protein that causes FSHD is regulated in a manner that's very intrinsic to the DNA. And chromosomes, of course, are encoding the DNA that encode every protein and lipid and metabolite that creates a human being and creates any living thing on the planet. And within each chromosome, there are stretches of DNA that encode proteins. They have very specific sequences, and those very specific sequences result in each of the detailed functions of a protein. But it doesn't just go from DNA to protein, it actually goes from DNA to RNA. And then from RNA, there's a transcription process from DNA to RNA, and then the translation process from RNA to protein. And this whole process is called gene expression. And you'll see in the next slide why I even started us here. And it's because when we look at the DUX4 gene or the DUX4 locus, it is controlled very tightly in healthy muscle cells. It is shut off and it's never intended to be expressed after the 2-cell stage of an embryo. So when we are all in our mother's uterus and from the 2 to 4-cell stage, DUX4 is expressed. It's got a very important purpose. Then it's shut off and it's never expressed in a healthy cell again. However, in FSHD, there are some changes to the DNA that can result in its expression. And the ways that DUX4 is shut off is by a method called epigenetic control. And I mention that because one of the therapeutic modalities that is going to be coming into humans, or is actually already in humans, is epigenetic control modality. And I'll speak about that when I speak about epic therapeutics. So typically the DNA is shut off by epigenetic control, and that's the librarian there saying, sorry, we're closed, so we don't get transcription, we don't get RNA, no translation, no DUX4 protein. And that is what is typically happening in a healthy muscle cell, and in fact, in every cell in our bodies. When we have FSHD, that control, that local epigenetic control is lost. It's no longer shut down there at the DNA, resulting in transcription of the DUX4 gene and then translation to the DUX4 protein. Then expression of the DUX4 protein, even at the exceedingly low levels that we can find it in patient muscle, is what results in FSHD. So how many ways, and the reason we showed you this entire slide is how many ways can we envision and can we therapeutically engage to shut down DUX4? And when I look at this slide, I become very hopeful. I don't know if any of you were able to listen to our CEO presentation that happened where Dr. Michelle Mellion and I were answering questions for the FSHD Society, and we were talking in detail about the failure of the Fulcrum drug losmapimod to reach its primary endpoint. And Michelle and I both commented that we remain very hopeful. And the reason we remain very hopeful is this type of slide. It's because there are so many ways that the therapeutic community is taking on to shut down DUX4. And it's really as a result of 30 years of research on the DUX4 gene. So we're still looking at the same paradigm, chromosome to DNA, and we can re-repress access to DNA so we can modulate the epigenetic control. One could correct the DNA sequence that blocks the expression of DUX4. We could move then from the DNA to RNA step, which is called transcription, and you could interfere with DUX4 transcription, or you could block or remove the DUX4 RNA. And some of the work from Charlie Emerson's lab that was presented earlier today will inhibit this step. You could also, going from RNA to protein, you could inhibit the translation, or you could block or remove the DUX4 protein. And honestly, I could stand here and talk to you about 1 to 2 to 3 companies who are working at each of these stages to eliminate the expression of DUX4 in muscle biology. And so I am, by nature, a very hopeful and optimistic person, but also because we have many shots on goal. I used to be a goaltender, so I loved shots on goal. So we have many shots on goal to take on DUX4, and I'm very optimistic, especially, and you'll be hearing a bit about the avidity data later. I'm very optimistic, especially as we've had some very beautiful data generated at avidity. So once we reduce or eliminate DUX4, there's other ways that we can also help muscles. And we're very actively, the society and others are very actively working to think about how we can stop muscle cell death, even with the presence of DUX4, how we can help muscle cells grow or regenerate. And I think this is very important because at whatever stage you're able to have the DUX4 turned off, if we can get those muscles to regrow or regenerate, that could give us a boost to some specific muscle functions. And then the reduction of fibrosis, and maybe the reduction or the accentuation of the immune system, depending upon where the disease is at, could be beneficial to FSHD patients. And these are all processes that are under active research and active therapeutic development as we speak. So therapeutic development is a long process, and I won't go into a lot of detail here, but the preclinical research that I was describing and also that you heard from Charlie's lab is all the work to understand the disease mechanism, how we can discover new novel therapeutics. So in clinical research is some of the preliminary work that helps us understand the natural course of the disease. And the reason we have to understand this is when we design a clinical trial, we have to understand how the disease progresses so that we know what to measure in a clinical trial and how to compare that to the typical function of a healthy muscle in this case. In the clinical trials, this is very essential. How do we have optimal, safe, and efficacious dosing? The FDA is very interested in the safety component. The therapeutic developing companies are very interested in safety and balancing safety and therapeutic efficacy. Efficacy against the disease and also demonstrating that it benefits the human being with the disease. So first you have to be very clear that you show that you're impacting the function of DUX4 protein in this case, you're reducing it, and that benefits the individual with FSHD. And then finally, this is a very important component that the society is paying a lot of attention to, and I think companies who are seeing some progress in this area are also paying a lot of attention to, and that is, does the FDA understand the disease in adults? Do they understand the disease in pediatric patients? Can we have them understand and agree that we have meaningful endpoints in both of those populations? They might be different endpoints in children or in younger individuals than they are in adults over 18. We have to convince payers of the value of the impact of the disease on life, and the patient community has been essential in coming to the FDA with the society and educating the regulators and educating the payer environment. And then longer term safety studies with medicines are going to be essential. I don't really want to go through this at all, except to tell you the phases are, they're not insignificant. Oftentimes a drug is tested in phase 1 for safety, and it can take up to 6 months. A phase 2 can take up to 18 months, and then a phase 3 can take 12 or 18 to 30 months. And the reason these times are so long with FSHD, and I don't have to tell you, it can be a very slowly progressing disease in some individuals. And because it's slow, the study has to go on long enough so that in your placebo arm or in the arm that receives the no drug treatment, the disease is seen to progress. If you don't have progression in that arm, you have nothing to compare your treated group of patients to, and so that's essential. So I will talk about this just a small amount, because the REACH study completed and they did not achieve a significance on their major endpoint, but they had developed, Fulcrum had developed losmapimod that blocked the transcription of DUX4 actually, so turned off the protein. And a phase 3 study was run, which failed to meet its primary endpoint, which was reachable workspace. It's an upper extremity endpoint that had been used in this study. And there was a lot of work done here. We were optimistic that it was going to be successful, and everyone was very disappointed. At the society and in the patient community, we've been doing a lot of work with Fulcrum, and they've agreed that they're going to share some component of their data with the FSHD Society, which will allow us to allow the larger community to interrogate that data and learn from it. There is so much data here. We have MRI data, we have reachable workspace data, we have everything that they measured, and we're hoping that it can be very valuable. It's probably the longest duration of measurement of FSHD subjects with some of these endpoints, and therefore very valuable. Roche has a different approach, which is not DUX4 specific, but it's actually, can we stimulate muscles to grow and develop? So can we go from inactive muscles to active muscles? And the mechanism that they've taken on, I won't go into much detail, but myostatin is a negative regulator of muscle growth, meaning it stops muscle growth. If they can kill myostatin by this technology, which is an antibody to latent myostatin, will you see in FSHD subjects that the muscles grow? It's been shown in other diseases in humans and in many preclinical models that inhibiting myostatin can result in muscle growth, so we're optimistic here. It's a year-long study, and I believe that's I think it's fully recruited and it's moving forward. Yeah, the study is fully recruited here and it's active. So we're waiting now for the readout of this work. I'm not going to speak in any detail about avidity, except that it's an antisense, it's an siRNA, sorry, to DUX4 mRNA. So it's targeting the DUX4 mRNA again, and they will, there's a representative here from Avidity who will speak about this, and I'll leave time for them to do that. And I believe this data will be addressed there, but this is the very exciting data that was reported on June 2nd at the IRC run by the FSHD Society, showing a benefit and also showing a reduction of DUX4 activity. So Arrowhead is another therapeutic company that's developing ArrowDUX4. They have a platform they call the TRiM platform, which stands for Targeted RNAi Molecule. Again, they're targeting the RNA for DUX4 for degradation using a complex technology, a series of chemistries that allow for binding of their molecule to the DUX4 RNA after it has been targeted to muscle cells. And I think the recruitment for this study is ongoing with a biopsy that will be conducted here with individuals from 18 to 70 and FSHD1. All right, and I already mentioned Epic Bio because they're taking on this novel approach compared to others, which is, recall that I mentioned that the DUX4 gene is very tightly turned off in healthy muscle cells, and that's a process called methylation or methylation of the D4Z4 domain. in FSHD subjects, the D4Z4 domain is dramatically reduced and the methylation is much lower, which allows for transcriptional machinery to come in, turn on DUX4 and allow it to be transcribed. What EPIC is intending to do and has shown some nice preclinical data that they can treat with EPI 321, and it remethylates the D4Z4, thereby shutting it down. And DUX4 is then turned off, and it re-represses access to the DUX4 DNA. What's unique and interesting about this is it's potentially going to be long-lasting, potentially even a one-time therapeutic. We're really looking forward to seeing more of this data. This work will be done in a number of countries, and it was believed to have begun in 2024, but I don't know if they've recruited their first patient, but we have a representative from EPIC here to talk with us. So these are the many more companies who are also working on developing FSHD therapeutics. So there's a lot of work that's going on. And I'm happy to take questions on that, but I also want to talk a bit about the natural history studies that the community is doing and how important they are. So we've had natural history studies ongoing in adults and in pediatric patients. We have another pediatric study starting this year with Dr. Jeff Statlin, and you can see actually that's Jeff here, who's done exceptional work with the Clinical Trials Research Network. The natural history studies are essential for us to be able to run good clinical trials so that we can define the measurements that are going to be meaningful, increase our chance of success, and provide better care for patients. So I think this is a bit of an advertisement. You know, if you meet the criteria for a natural history study, especially in the pediatric sphere, you'll be asked to attend at least 3 studies, perform strength and movement. I believe there will be MRI, and I believe there will be biopsy also, blood, saliva samples taken. And I think you were able to see from some of the data you saw today, but also in understanding the endpoints that are measured in a clinical trial of a new therapy, that taking these samples is essential to our success. And I wanted to show this slide because it shows all the places in the United States and Canada where those clinical trial natural history studies are happening. MOVE Plus, and the pediatric study. The individuals there's one individual who's really exceptional. There's many exceptional people who work with Jeff, but Michaela Walker is the project manager, and she can be contacted if people wish to be involved in those studies. And I don't think that I'll talk about that. I talked a bit about that. So I thank you for your attention, and I'll take any questions if there are questions about what we presented. Okay, well, we're going to the question there.
I just saw that, I just walked in, I saw that slide that you had that showed all different companies.
Yes.
Wow.
Yep.
That slide have looked like 5 years ago.
Oh. 5 years ago, you would have seen 2 or 3 companies here. Fulcrum was really moving, and I have to honestly say, I worked for Fulcrum for the 5 years that we developed that molecule. We were pretty much working independently with 1 or 2 other companies at the same time to develop an understanding of the disease in a way where we could run clinical trials. Yeah, so I think that once the genetic tests, and we should just reiterate that having a genetic test that demonstrates very clearly that you do have FSHD is essential for participation in the natural history studies and in the clinical trial setting. And they were not easy to be run even 5 years ago. They were very complex diagnostic tests, and those have become much more accurate, much more simple, and much more reliable. So there's many things that have changed since then. Science has moved dramatically rapidly in the 5 years to bring us all the different approaches that these companies are taking. Yep.
So earlier, when there was a discussion or explanation of how the research is being done here, and was it lipid, is that?
Yeah.
Okay. Being potentially simpler, less expensive, more easy to produce, but they didn't have a company behind them.
That's right.
So what kind of effort is involved in getting that relationship between the research that's being done and a biopharmaceutical company?
Yeah, and it's enough preclinical work must be carried out to convince a biopharmaceutical company that the approach has the possibility for success in humans. And as Dr. Emerson's lab was describing, there have been studies in rodents, I believe it was in mice, I don't think it was in rats, that showed that they could successfully target muscle with those lipid molecules, and that there was no immune response and very few deleterious effects. But there haven't been any treatments with those specific lipid molecules yet in humans. So the first thing is enough good preclinical work like that work to show, and often it's done in rodents, sometimes it's done in dogs or in pigs, to demonstrate that the concept works. It gets to the muscle, it reduces DUX4, and it doesn't activate the immune system, typically one of the most deleterious effects we could see. Once that preclinical work is done in a number of animal models, oftentimes you'll have a therapeutic company that's interested in picking it up. And I know that there is some interest.